Good day, everyone. It's a pleasure for me to talk to you on one of my favorite topics, the gut microbiome, especially in early life. And I will present today a little bit about the importance of the microbiome in early life, but also biotics or nutritional therapies to support microbiome in order to support health in early life. I'd like to start with a disclaimer for full transparency that I have two positions. I'm a senior director at Knol Nutrition Research, but I'm also special professor of microbiology at Wageningen University in the Netherlands. Agenda, so I will really focus on the microbiome, on the microbiology of the gut, and in how this supports immune fitness, but also overall healthy development, and nutritional strategies we have to support gut health and the microbiome in early life. And I will end with some key messages. I guess everyone, all of you, are very much aware of the big news around the microbiome that started, I don't know, 10, 20 years ago, where we started to realize how many microbes we have inside of us and how important they are actually for our health. And this goes beyond just digesting our food. It also is indeed a critical metabolic organ. The gut microbiome has a huge immunological function, and it is high numbers in the gut, where, of course, the surface area is huge. So there's a constant interaction of these microbes with us as humans, and that is essential also for health. Large numbers, and due to all kinds of new technologies, we start to understand the complexity, the diversity, but also the huge functionality that the microbiome can harbor, and that can help us to digest our food, to produce vitamins, short-chain fatty acids, but also to give immunological signals and to keep a symbiosis in a homeostatic way. And links have been, of course, made with many of these benefits and functions of the gut. I'm sure you were aware of some of these. Of course, microbes can only digest fibers, polycarbohydrates that we cannot digest as humans. Many of them the microbes love. They can still degrade them, use them as carbon sources, energy source, and often produce short-chain fatty acids from the metabolism of these fibers that we then as a host again use to support healthy epithelium, but also as an energy source for certain types of cells. The microbiome can produce vitamins, ones that might not be per se in our diet, but the microbes can then, as metabolites of their own metabolism, produce vitamins that we can then also again use. Of course, the microbiome, with all its large capacity and cell numbers, play an active role in metabolism by digesting our food, making available certain nutrients, but also supplying us with signals, regulating appetite, regulating metabolism. Of course, the microbiome defends us against harmful microorganisms, something called colonization resistance. So it's quite hard for an undesired bacterium to colonize the gut if you have a healthy microbiome. We see that, for example, if you use antibiotics, you disturb the microbiome, then, for example, C. diff can colonize. But normally, when the microbiome is stable and healthy, then it also provides a barrier against the invasion of pathogens. And of course, these microbes are still a very strong immune signal. So the immune system senses, of course, the lining of the gut. 70% of the immune cells at some point are in the gut. So this is a great interface also for immune interactions. And of course, bacteria are a strong immune signal. So also there, the microbiome provides all kinds of signals that impact the immune system, as also do the short-chain fatty acids they produce. We now know that butyrate, for example, is important also for development of T regulatory cells in the immune system. So also there's a constant crosstalk between the host and the microbes. And maybe the newest part of the science in the microbiome area is this gut-brain connection, which I think is fascinating, where we see, well, mostly now in animal models, but also some first human data, that microbes in the gut can actually impact the way we feel, our mood, or even how animals, for example, behave, dependent on the composition of the microbiome. So very interesting and important links between the microbiome and our human physiology and human health. And this is, of course, particularly important in early life because when a baby is born, the gut is still relatively immature, naive. It still has to develop. It's not fully functional yet. And the introduction of food, the introduction of microbes into the gut is also an important signal for the gut to develop. So after birth, a few steps still have to be made that are important for digestive capacity of the gut, for the immune function in the gut, but also to set up this gut-brain axis crosstalk. And of course, within all of that, nutrition is important, what we feed a baby right from birth onwards, but also how the microbiome in the gut develops to steer also the gut maturation in an optimal way. And this is, of course, quite intriguing. In the womb, the baby is still protected from microbes. There's a bit of discussion in the literature whether we're already exposed to microbes in utero. Maybe we are, at least to some signals. But of course, the numbers of live bacteria is very low in the womb. And we're actually colonized when we enter into a microbial birth at the moment we are born. Microbes are everywhere in the environment. So immediately these microbes will start and colonize the body, the skin, but also the GI tract and all the niches that are available for microbial colonization. And also here we see that breastfeeding is really important for, of course, growth of the baby, but also to steer this process of microbial colonization. So breast milk also has evolved as an optimal nutrition, not only for the baby, but also for the developing microbiome, and steers also this process of obtaining the right microorganisms in our human body microbiome. And we see that also, for example, in the colonization of specific species. So breastfeeding, we now know, is really stimulating, for example, the colonization of bifidobacteria. Because in human milk, we have these human milk oligosaccharides that are very good substrate for specifically these bacteria. So this is an example, I think, of coevolution. We as humans, we produce something in our milk that is liked very much by bacteria that then colonize the gut and then help also a healthy gut microbiome and gut health development. And this is quite a dynamic process, of course, in early life, from an almost sterile environment to the rapid colonization of these bifidobacteria that then start to produce metabolites that also other bacteria again can use. And in early life, the first days, first weeks, we see a rapid colonization and development of the gut microbiome. But of course, in modern life, we now also know that there is quite a few challenges to this developing microbiome. Normally, the microbiome would come for a large part from the mother. There's exposure at birth, during delivery, and microbes will also go from mom to baby. And then depending on development, on diet, the microbiome will further develop. But there's quite a few things that we introduced or that happened in the last, let's say, 100 years that can significantly impact this sensitive development of the microbiome in early life. Mode of delivery, for example. We know that C-section born babies, they really have a different colonization, a delayed colonization, because they're exposed to microorganisms in a very different way. Early life antibiotics is another example. Antibiotics, of course, a great invention that saved many lives, but it's also used quite a bit already in early life. And you can imagine that when the system is still establishing, that antibiotics can have a big impact on the developing microbiome. Also, preterm babies, we know the gut is also not fully mature there, and that they also have an aberrant gut microbiome development. And preterm numbers can be quite high and seem to be still increasing. Also, environmental exposure, I think, has changed quite a bit in the last, let's say, 100 years. Mostly hygiene, of course, and how we deal with our environment and with disinfectants. So also there, the exposure has changed. And then lastly, also maternal factors may impact this early colonization, because if a mother has already a compromised microbiome because of a disease or an illness or antibiotic use, of course, also the mother is transferring potentially different types of microorganisms to the offspring. So these are all kinds of risks for the developing microbiome that have significantly changed in recent uh, era of humans. And why is it so important? Not just for, let's say, the microbes that colonize the gut, but also for us, the host site. Um, well, I think the first 1,000 days are indeed a very critical period for the development of all kinds of organs and tissue, where the gut is also an extremely important one, of course, because it has to be the place where, for the rest of our lives, we digest our food, we deal with allergens and antigens, we have to absorb the right things, we have to get a symbiosis between the microorganisms residing in the gut and us, without having too much inflammation, but with having regulated response towards antigens and food. And this starts from very naive guts when we are born, but then rapidly, of course, when we introduce microorganisms and food, the gut also develops. It's trained, it's learning all kinds of tricks for digesting of food, but also to initiate an appropriate immune response. So there's a constant crosstalk also between the developing immune system and the entering microbes that then decides the composition of the microbiome, and also how the immune system is trained in this important interface in the gut in early life. And the interactions, I think, are many and very important ones. Of course, there's a competition between healthy bacteria and maybe potentially pathogenic bacteria in the lumen. There's all production of all kinds of molecules that impact this interaction. Then these microorganisms can interact with our epithelium by exchanging metabolites, but also with direct signals that can be immunogenic. Important cell types are, of course, the epithelial cells, but also immune cells, like dendritic cells, that even seem to sense what's there and to then react in an appropriate way. And how this initial exposure is happening also is very important for the way the immune system develops, whether T regulatory cells are developed, or whether the Th1, Th2 balance is also developing in the right way, and whether Th17, for example, is yes or no stimulated. So very important phase where, yeah, it's really training, I would say, of the mucosal immune system and gut health that is driven very much by the entering food, but also developing microbiome, this stage of life. Well, multiple factors can impact, as I already mentioned, many of them, preterm C-section delivery, antibiotic use, which can all lead to what we call dysbiosis, so a suboptimal development of the microbiome. Normally, in a healthy development, no disturbances, and this will also optimally support immune tolerance, gastrointestinal homeostasis, and a healthy metabolism. But if the microbiome tips over and becomes a dysbiosis, then this is also correlated with immune diseases like allergy and asthma, gastrointestinal diseases like IBD, but also metabolic diseases like obesity and diabetes. So what is dysbiosis? It is a term that's difficult maybe to understand, but what we believe is that a healthy microbiome is indeed balanced and diverse, many different types of bacteria, symbionts, commensals, and also pathobionts, which is a mix that we all potentially have, but normally the body is well equipped to regulate the composition and activity of these different types of bacteria. And then the whole system is in an immunological balance, and we have this immune resilience. We can react and regulate this system. However, if the gut microbiota dysbiosis becomes too large and the undesired types of bacteria become too dominant in their presence or activity, we can tip this balance, and this can then also lead to immunological dysregulation or even non-specific inflammation. And especially in early life, when the system is still establishing, this can, of course, have an important impact. And these imbalances in the gut microbiome have also been linked to many health and disease issues, like asthma and allergy, but also infections, obesity, metabolic disorders, also infantile colic and functional gastrointestinal disorders being linked to disturbed microbiome, as also inflammatory bowel disorders, of course. But now also we see evidence that this may be linked to aberrant behavior and autistic spectrum disorders. And an important one, I think, is allergy. There's a lot of evidence now also out there that shows that it's important to establish a symbiotic relationship between the microbes and us that develops then also tolerance to antigens in early life. And there's a lot of evidence from animal models and also demonstrations that if you manipulate the microbiome, that you can indeed prevent allergic diseases and also autoimmune phenomena. And this is important in early life, but we also think, because the first 1,000 days are so important for programming of health, let's say, that we should also keep this in mind for the health when we're adults, that this process is developing in the right way in early life, because it's also important for later life. So what nutritional strategies then do we have to also support the development of the microbiome in early life? And of course, we have learned a lot from human milk, and we're still not done yet, I think, studying human milk, because it's so complex, so rich in components, but also in functionalities. There's cells in there, there is oligosaccharides, not so much even used by the baby, but really in there because of feeding the microbiome. We have bacteria, bacterial compounds in human milk, all kinds of complex fatty acids and lipid structures, of course, all the nucleotide, vitamins, minerals, micronutrients that also we need to feed the baby, fat and protein as macronutrients as well. But many of these, they have also functional properties that are important for the baby to grow in an optimal way. But many, as you can see already here, in there also to steer the microbiome development, as is the prebiotic fibers in human milk, the bacteria and bacterial products that are clearly there to steer the microbiome. Let alone, of course, there are still also the antibodies that against pathogens, antimicrobial peptides. So even there on the protein fraction, there's also molecules that can steer the microbiome development and regulate the right microorganisms to colonize. Just to focus on that is to show that human milk is not just feeding the baby to grow, but also feeding the microbiome to develop. We now know in human milk that there is live bacteria and that there is oligosaccharides, prebiotics to feed microorganisms. The live bacteria, I think, is now well established that these are indeed there, that human milk is a niche for the colonization of bacteria like bifidobacteria and lactobacilli, and that these are constantly found low numbers, but they're still there. And this, of course, is also a way to feed and inoculate the baby with microorganisms. These numbers are low, 10 to the third, 10 to the sixth of bacterial cells, and quite often we see streptococci, bifidobacteria, types of bacteria that we also see in the early days of colonization in the gut. And we anticipate also that these bacteria play a role in the rapid establishment of a microbiome, and that these are important also against infections, but also contribute to the maturation of the gut and the gut immune system. In human milk, you also find all kinds of compounds produced by bacteria. Quite interestingly, there is recent data where people have looked with sophisticated analytical technologies into the details of the composition of human milk, and we see all kinds of molecules that we as human beings, we cannot produce, but that we think are directly derived also from microorganisms. So these bacteria are there, they sustain, they are active, and they produce also metabolites that then end up in human milk. So human milk also contains bacteria and their products. Also, of course, the HMOs, the prebiotics, this is just a schematic overview of all the molecules that we can find in human milk. It's complex, it's quite interesting, complex sugar structures that may have different functions, but one function is also, of course, that bacteria like them. They can use them as carbon and energy source, degrade them, ferment them, and use that to grow and prosper also in the gut. And specifically on the left, we see that bifidobacteria, but also Bacteroides, for example, are very much specialized on using these kinds of glycan molecules. They have all the transport systems and enzymes to degrade these molecules. So again, I think this is clearly a co-evolution of humans with specific types of microorganisms. We produce an optimal lunchbox for certain bacteria that then help to colonize the gut in an efficient way. And the level of these molecules is indeed very high. It's 3 to 15 grams per liter, which is more than there is protein, for example, in human milk. So a lot of food for microbes also in human milk. But there's also new opportunities, I think, to modulate the gut microbiome. Prebiotics, already mentioned, so substrates for beneficial microorganisms. Probiotics, I'm sure you're familiar with these terms, where you actually in your diet use or eat, consume live bacteria, also with the intention to provide health, optimal colonization or improved gut microbiome. And the last one, maybe less familiar, is the postbiotics, which I will also come back to. These are the official definitions. I will not spend too much time. Maybe just the postbiotics, I think is quite new. And postbiotics are bioactive compounds that are produced by food-grade microorganisms by fermentation. So this is conversion of a raw material, let's say milk, fermented by bacteria, that then leads to all kinds of bioactivities in this fermented product, which we then call postbiotics. And this is also a concept that we use in infant nutrition, for example, to provide the infant with microbiome signals to optimally stimulate also the gut development. Just a few examples on the human milk fiber or human milk oligosaccharides and the prebiotic concepts that we see also we can use in infant nutrition. Big example already out there for a long time is this mixture of galacto and fructo oligosaccharides. Well established and very much inspired by human milk, where we see these thousands of structures of human milk oligosaccharides, different types, different lengths. And with the GOS-FOS mixture, the aim was, of course, also to have a substrate that supports the growth of beneficial bacteria, but also had to have a bit of this complexity, the variance in molecules, size of the molecules, to really feed microorganisms in the developing microbiome in an optimal way. And by now, of course, we have studied this prebiotics to a great extent and with a lot of evidence, clinical studies, and publication supporting indeed the fact that we can stimulate the growth of bifidobacteria, for example. That we can make the microbiome much more similar as human milk fed infants. So the microbiome composition, also microbiome functionality, we can really impact with prebiotics, because cow's milk is the base for many of these formulas, does not contain fiber, but by adding these prebiotics, we can indeed feed the microbiome and stimulate it in a very interesting, beneficial direction. So a lot of data, I will not go to all the details, but we have, of course, different benefits demonstrated, like stimulation of bifidobacteria, microbiome closer to breastfed infants, but also supporting a gut environment with typical short-chain fatty acids, reducing also colonization of pathogens, for example, in preterms, but also higher SIGA. So these are all documented effects of using prebiotics in early life. Here's one example. We have some proof behind the reduction of infections, but also the use of antibiotic courses. So indeed, if you improve the gut microbiome with prebiotics, you get more bifidobacteria, you get less pathogens, and you seem to also need less antibiotics in early life, which is, of course, a good thing, because the antibiotics are quite an impact on the microbiome, but also have a risk of introducing antibiotic resistances. Also, and this again shows the link between the developing microbiome and the immune system, is that we see effects on atopic dermatitis in the Moreau study. These are infants which were at high risk of developing atopic dermatitis. And we see that if you use the prebiotics, we can indeed improve the gut microbiome with a significant increase in bifidobacteria. But we also see a reduction of the cumulative incidence of atopic dermatitis. By targeting the microbiome, we can also see an effect on immune development. And interestingly, again, coming back to the programming of health, although these infants only received the formula in the first months and year of life, we still see this effect persisting also in the 5-year follow-up. So it seems to be important for early life, but also these effects can persist in later life. Then a few words on probiotics, which is not just using the substrate for the microorganism, but also using the live bacteria themselves in nutrition. An example is the Bifidobacterium breve M16V, where there is a lot of documentation behind this specific strain, because we all know that probiotics are very much specific. You cannot talk about probiotics at large. You really have to specify the specific strain, the specific benefits of this strain. And we have focused a lot of our work on the breve M16V, which we started screening, I don't know, already 15 years ago, I would say, from a cultural collection that we had, our own strains and what was available at the time. And we benchmarked those for all kinds of desired phenotypes, safety profiles, and also in preclinical screening, we benchmarked many of these probiotic strains to identify the most suitable candidate for use in early life. And we ended up with this breve M16V, which is in all kinds of aspects superior, because it behaves well in infant formula, infant nutrition products. It has a very good safety profile, and it's also a typical infant species. So breve can use human milk oligosaccharides. It's quite resistant against the antimicrobial activities in human milk, so it's very compatible also with human milk. And breve is also a species that we see developing rapidly in healthy breastfed infants. So it's very close to a human. It's also isolated from a breastfed healthy infant. It's commonly isolated also, breve, from breast milk. That's extremely relevant, I think, this species in early life. And again, safety, of course, is extremely important, because some of our infants, they may be less healthy, like preterms or C-section born infants with some health issues. Of course, we need to be then very careful about the safety profile of the probiotics that we use. And M16V was screened, the genome was sequenced, and we have a very good safety profile of this probiotic. And it has also a long history of safe use, because it's used in preterm infants already for a very long time. And this specific strain also has no particular antibiotic resistances, for example. And also from all the in vitro and in vivo work we did in animal models, we see a very good safety profile of this probiotic strain. Of course, what we now use quite often is this combination of prebiotics, which have a more general impact on the microbiome, but also specific probiotics in something that we think can bring synergies, and something that is called symbiotics. And we were especially intrigued also from the animal work we've done in some immune models. We clearly saw that if you combine the substrate for microorganisms, but also the live microorganisms in a symbiotic combination, we quite often see better effects on outcomes. And we have tested this mix of symbiotics in, I think, a very relevant group with C-section born infants. This is also a study recently published, a Julius study in Asia, where we, we of course know from the literature that these C-section born infants, they have a compromised microbiome development. That's been published several times. And what you often see is indeed this delayed colonization of beneficial bacteria like bifidobacteria. But we see in this case that with the symbiotics, already after 3 to 5 days, we see that also we can correct this bifido gap. So with the use of the symbiotics, with the breve M16V, we see a high level of colonization of bifidobacteria in early life, and also better short-chain fatty acids, better gut microbiome development in these C-section born infants. We also looked not as a primary outcome, but more as adverse events, also at some immune outcomes. And to our surprise, we also saw indeed that we saw improvement in the skin disorders and atopic dermatitis that we quite often see in these type of infants. So I think a very strong example where a severe gut microbiome disturbance, C-section, can be corrected with a nutritional concept like symbiotics. And maybe the last one, the postbiotics, also to mention those. So they're not really the direct substrates for microorganisms in the gut. They're also not the live bacteria, but they are the metabolites and all the direct, indirect signals that microbes can give. So postbiotics, again, these are the bioactivities, the compounds that are produced during fermentation. So it's really bacteria being active in a matrix. And these postbiotics may positively also modulate, of course, the gut and also the immune system. As I said, also in human milk, we find all kinds of products and metabolites of bacteria. And we also can, of course, mimic that by producing postbiotics. Of course, it's also complex because what is really the active moiety? It can be many things, because if you have a matrix with microbial strains, you have, of course, proteins, peptides, lipids, specific carbohydrates, vitamins, but also the organic acids, short-chain fatty acids, cell wall components of the strains you use that could be immunologically active, but also all kinds of complex molecules that are produced that could all be considered postbiotics, and that then could bring also benefit to the infant. So again, not all postbiotics are the same, so it very much depends, of course, on what you use to ferment, but also the specific bacteria that you use to produce these postbiotics. And this is also an innovation we developed in recent years. The fermented formulas were already used for a long time in France under also the name of lactofidus, where we ferment milk with also a bifidobacterium breve and a Streptococcus thermophilus. But then we combine this now with also the prebiotics, the GOS-FOS, because this also then provides a substrate for the microbes. And we've studied this, and this is, I think, a unique concept, combining the prebiotics and postbiotics to bring also, without using live bacteria, still benefits to the microbiome and the gut in early life. Well, we have also here, we had a careful selection, and I will also not go into detail, but these two strains were very much selected from also a large collection for potential superior characteristics in the process of producing postbiotics, but also in producing molecules and signals that are relevant for healthy gut and immune development. So they're considered also safe. They're on the favorable list also of EFSA. And also these type of bacteria, Streptococci and bifidobacteria, occur in human milk. Well, we have also, from the postbiotics, from the part of the fermented milk, we also have evidence, clinical outcomes from the past, clinical benefits. So we see increased poliovirus antibody response, for example, less severe diarrhea, but also upregulation of fecal secretory IgA, for example. So this also shows that you do not always necessarily need the live bacteria to also have this immune stimulatory effect. Of course, to really colonize the gut, you need live bacteria, but also the postbiotics, the metabolites, and the dead bacteria can indeed trigger a healthy gut development and immune development. So now, by combining the two, the prebiotics and the fermented formula, the postbiotics, we have a lot of evidence that indeed these formulas are safe. They support healthy growth. And actually, from the studies, we see that these formulas are very well tolerated. So that's one of the benefits of these fermented formulas, that they are very well tolerated and give very little gut issues. We see that the stools become closer again to human milk-fed infants. And interestingly, we see that we have a lower incidence of investigator-reported infantile colic, also showing that these fermented formula, these postbiotic formulas, are very well tolerated and seem to be effective also in maybe prevention or treatment of infantile colic. So we also studied, and we're still studying these concepts, and we see to explain what happens and to also even improve our formulas more. We see that using this combination of prebiotics and postbiotics, we see that the microbiota composition and activity is even closer to breastfed infants than by using the prebiotics alone. And this could be beneficial, of course, for the gut and immune system. We get more and more data on the microbiome, also using new technologies as the new sequencing technologies, for example. And we see, again, higher bifidobacteria, but also, for example, lower levels of C. difficile, and also an overall microbiome profile looking at all the 16S data, which is much closer to breastfed infants. And also the microbiome activity is interestingly changed. We see still further improvement of the microbiome activity, which I think is maybe even more important than just the taxonomical composition of the microbiome, because it really is important what the microbiome is doing in the gut in early life. So just to wrap up and finalize, and I think in the symposium you will hear much more about some of the concepts that I have already started introducing here by other speakers. I will just finish with some key messages and very general ones. I think you all agree and know that the gut microbiome does play a very important role in our health. It's an important organ consisting of many cells and that help us to remain healthy and to deal with our food in an optimal way. It's also quite clear that early life nutrition and human milk being the most, of course, suitable and important nutrition in early life, that is a key factor for gut microbiome development, but also functionality of the microbiome. But now today, of course, by understanding more and more how this interaction, this interplay works, we can also design, study, and use concepts, the biotics like pre, pro, syn, or postbiotics, and that they are quite well investigated now and proven nutritional strategies to support the gut microbiology, let's say, and the gut microbiome, and that this also and can help to support infant health in infants that have a compromised microbiome, but also, I guess, in the general population where still this gut, of course, needs the right support, because when we're born, it's still a relatively naive system that needs to receive the right nutrition to support a healthy gut development and a healthy overall development of the infant. I'd like to end with thanking a lot of people, my colleagues in Utrecht, but also in Singapore, and the collaborators and my colleagues at the Wageningen University, of course, but mostly, of course, the pediatricians and parents that participated in all the studies, because I think this is really important. We cannot do it in the lab. We really have to see and study how this also then works in infants. So the human studies are really very important. So just a reminder, health through gut, we don't only feed us, the humans or the baby, we also need to keep in mind that we really have to feed the microbiome in the most optimal way. And with that, I would also like to thank you for your attention, and I hope that you enjoy the rest of the program.